let's get started. Hopefully my multimedia will actually work this time as opposed to the last time when it failed abysmally. Wanted to quickly finish with adenoviruses. Just a review, as usual. They have these amazing structures. Oops, back up here. Still working on this technology, of course. Amazing structures with the pseudo T equals 25 symmetry and sort of the classic icosahedrally symmetric virion with these penton bases and fibers. And this is why people like this so much, as sort of the stereotypical idea of a virus. Entry is really quite different for these adenoviruses because, again, they're naked viruses. They don't have membranes, so they can't fuse easily. They have to get inside the cell through interactions with receptors, again, not surprisingly, sitting at the ends of the pentons here, the knobs, the Coxsackian adenovirus receptor, comes in through receptor-mediated endocytosis. Endosomes, of course, become acidic. That activates viral proteases, which start to break down the virus, and then that can escape from the endosome. That actual escape is still not a completely understood process. Then the partly degraded particles get transported to the nucleus on the cytoskeleton. Once they get there, another viral protease will break down the rest of the structure, release the genome. That genome, of course, is now a linear double-stranded DNA, unlike the circular ones that we were talking about before. We'll see another weird kind of DNA formation today in the Poxviruses. Once it gets in, then you have immediate early transcription, which are our friends E1A and E1B that mess with the cell cycle, mostly trying to get it to work, again, through activating mostly RB, which turns on the E2 factors, which are important for making the E2 genes, and oh yeah, that other boring stuff that the cell normally does in terms of the cell cycle. These guys are all involved in replication, and most of the replication in these adenoviruses is going to be viral proteins. So viral RNA polymerases, sorry, not viral RNA polymerases, viral DNA polymerases, viral terminal protein, because that's what the primer is for replication. You've got a viral single-stranded DNA binding protein, which basically serves as the helicase. So it's really all the viral proteins that are involved in replication. On the other hand, the transcription is almost all going to be done with cellular proteins. So it's the cellular RNA polymerase, cellular proteins that are involved in splicing, cellular proteins that are involved in tailing, and a lot of what we know about eukaryotic transcriptional initiation, splicing, tailing just comes from the study of these adenoviruses. And then at the very end, we talked a little bit about therapies, and because we know so much about these adenoviruses, and because most of them don't cause much in the way of disease, although there was actually, people see the news reports, I think it was in Maryland, adenovirus type 7, that unfortunately somebody died from some disease there, and all kinds of issues about that. But in general, adenoviruses, and particularly adenovirus type 5 and adenovirus type 2, are really good in terms of viral vectors. So bringing your favorite DNA inside a cell or causing a cancer cell to die. So any questions on the adenoviruses before we move on and talk about the poxviruses? Okay, so the poxviruses, sort of our progression now. We're getting bigger and bigger and bigger. Of course, on Friday, we're going to get really tiny. But here, these are the largest DNA genomes, and DNA genomes in general are larger than the RNA genomes for viruses. These, as we'll talk about right at the end of the course, are part of the nuclear cytoplasmic large DNA viruses. Again, we'll talk more about that when we talk about the giant viruses. The first of these were the poxviruses, and they do seem to be related to those. These have linear genomes. Linear is in quotes, which anytime Stedman puts something in quotes, it means that it's not exactly linear. You can think of these as being linear with ends, like you have in the adenovirus genomes. But here, these ends are actually closed ends. So you could either think of this as a linear double-stranded DNA virus with closed ends, or a single-stranded DNA virus that's completely complementary to itself. Just two different ways of describing it. We'll talk more about that a little bit later on. Larger and more complicated than Herpes viruses, obviously I didn't update this slide from last year. We didn't talk about herpes viruses. There's a nice chapter in the textbook and then a lecture from last year. If you're interested more in herpes viruses, happy to talk about them. 10 weeks is not enough time for this course. But definitely in terms of adenoviruses, much larger, many more proteins involved, and partly because of that, and again, this whole idea of progression from the adenoviruses, which have all their replication proteins, now these poxviruses also have all of their own transcription proteins. And so we're getting more and more autonomous, more and more cell-like in terms of the virus, rather than what you have in some of the really tiny ones, again, starting out with the polyoma viruses, which are really very, very dependent on cellular processes. Here, these are very independent of cellular processes. Why do we care about poxviruses other than the fact that they're really interesting genomes and really interesting regulation? Is because smallpox was probably the nastiest virus disease that anyone has ever known, way beyond things like HIV or even for that matter, flu. And partly because of that, they're really good vaccines, and we'll talk a while about vaccines, and that's my extra little video a little bit later on. Smallpox has been completely eliminated, at least from natural circulation, which is a real, I think, amazing accomplishment of public health. So we'll spend a little while watching the video and then talking a bit more about that. And then just because these viruses are really autonomous, they have all of the machinery that they need for making more of the genome, making their RNA. They're a great source for getting lots and lots of messenger RNA. And so a lot of the studies with messenger RNA earlier on had to do with that. So what are our key concepts for, now of course I can't get them to advance. Far too far along here. See, it's technology, it's not working for me. So where do they come from? And we'll talk a little bit about disease, and again, we'll talk more about vaccination at that point. And then what the genome looks like, again, this strange kind of linear closed covalent ends or double-stranded linear. How that gets replicated is bizarre because of these end structures. And so because of the bizarre end structures, it's not surprising that you're going to have a slightly different kind of replication going on. Transcription, it is its own viral machinery, but because it's its own viral machinery, well, actually not necessarily because, but the viral machinery is actually really quite different than the cellular machinery in terms of how the messenger RNA is being made. So they're viral proteins, and specifically the viral transcriptional regulatory proteins are really quite different. And then how they get outside of the cell is really quite interesting because they have these really interesting structures. So this doesn't look either icosahedral or helically symmetric, kind of blob-like, but very consistently blob-like, and has, in some cases, one membrane, and in other cases, actually even two membranes around the outside. So we'll start talking a little bit about viral disease. Smallpox was known well before viruses were known. Again, not unlike polio. There are some of the very first written records or pictorial records really, have indications of this kind of disease, very distinctive in terms of the pox which form on patients. So thousands of years ago, smallpox, the Latin name is variola, and actually confusingly variola major, so the major variola is actually smallpox. Big pox is syphilis, just in case anybody was wondering. The reason that most of us have very light skin, probably, living in this continent, probably has to do with smallpox. Because when the conquistadors came and were battling the indigenous peoples here, smallpox did away with many more of them than the conquistadors were ever able to. And so probably that's why most of us have light skin rather than considerably darker skin. Smallpox has about a 25% fatality rate in adults and much higher in babies and young adults. So really nasty disease. Turns out that it's not just smallpox. There are quite a few different pox viruses. that infect different animals. Many of them actually don't seem to get anywhere near as sick. And so why smallpox is so nasty and these other pox viruses less so is not completely understood. And that's partly why people are continuing to study some of these other pox viruses in terms of understanding what's going on with smallpox. These are just some of the pox viruses. Monkeypox is actually current issue, particularly in Africa. There are spontaneous outbreaks of monkeypox. Has not quite as high a fatality rate, but still about a 10% fatality rate. Seems to mostly replicate in rodents, in this case particularly squirrels, but can replicate in humans and monkeys. Big advantage as far as getting rid of smallpox is that the reservoir host for smallpox is just humans. It doesn't seem to infect anything else. And so that's basically why we're able to get rid of it. Molluscum contagiosum, another pox virus, but seems to cause very little in the way of disease other than these sort of pox, raised kind of lesions that you get. Otherwise, Vaccinia is interesting because we really don't know where it came from originally. There are a number of discussions about where Vaccinia came from. Was supposed to come from cows, but there is actually a cowpox, which is not Vaccinia pretty clearly. Some people say that it's more like a horse pox. Basically, Vaccinia was what came through vaccination and has just been isolated literally over hundreds of years. So where it originally came from is still an open question. It definitely can infect humans and cows, but where its reservoir host, at this point, the reservoir host is probably in the lab because that's where people are actually making it. And then, various numbers of others of these as well. Even insects are infected by pox viruses. But because you had such a high fatality rate, people have been very interested in how to deal with smallpox. And one of the things that was noticed quite early on was that if you'd been infected by smallpox and you didn't die, one of the 75% or 60% of the lucky ones, then you were resistant to being infected again. And so you could be around someone who was highly infectious and not come down with disease. So this process called variolation. So remember, variola is the name of the virus. So variolation is just taking a small amount of virus and infecting someone with it. The death rate from that was only about 1%, and of course, no one will be able to get that through a clinical trial these days. But 1% death rate's a heck of a lot better than 40, or for that matter, 25. So this was done quite a lot in India and China. There are all kinds of stories about people taking the pox off of someone who was infected and grinding it up and then spraying it into people's noses. Apparently that worked. Again, interesting ways of doing things, but it seemed to work very well. But that was still had this 1% death rate, which was fine. It was a lot better than, again, 25 or 40%, but still not optimal. And there are lots of discussions about how the news came to England, that this is what was happening. In fact, people in England were in fact doing this at around the late 1700s. But then Edward Jenner, who some people have said is sort of the hero of public health, came up with this hypothesis that the milkmaids who weren't getting sick from smallpox, they might actually have a disease that could protect against smallpox. So he did an experiment, which again never would be permitted under current rules, where he then had a child who hadn't been exposed to smallpox, gave them some cowpox, and then challenged them with a variolation here with smallpox, and found that this person, the name I'm forgetting now, but was then resistant to disease. And there's, say, 14th of May, so close to the anniversary here. I think I posted a couple things on Twitter about this, where the first experiment was done. He published it a couple of years later, and it was and that should be cowpox. is giving immunity to smallpox, called vaccination, because vaca is the cow. And that's where Vaccinia comes from. This process was used all throughout England. A lot of it was sent to the US. Thomas Jefferson was a big fan of vaccination. And one of the ways that apparently the virus was transmitted is someone got vaccinated and then got on a ship and then went across to the US, and they got one of the scabs from where they'd gotten the pox inoculation and then used that to infect other people. And so the vaccination process. So because of this vaccination, vaca has worked extremely well, and because smallpox only infects humans, it was basically eliminated in 1978, and we'll see the video about how that happened. And people said, well, this is this really nasty disease. We want to get rid of it. There's no reason that we want to hang on to smallpox anymore. And I think it was in '96 they were supposed to have, there was this big arrangement. At that point, there were 2 known sources of smallpox, one group at the CDC, one group at the Russian equivalent Vector, and they were going to have people from the different agencies go to each different place. They're going to turn on the autoclaves at the same time, push go, and all the smallpox was going to be gone. And then they decided that maybe, just in case, we might want to not do this for a couple of reasons. And the first one was, yeah, we've got Vaccinia that works pretty well, but we don't really have any good antivirals. And because smallpox is different than almost all of these other pox viruses, maybe we want to keep a little bit around for some experiments. So there actually are continuing experiments going on with live smallpox at the CDC and at Vector I think it's in Siberia, if I remember correctly. This is all being regulated by a group called the WHO Advisory Committee on Variola Research. This person is not included in that advisory committee. May look vaguely familiar here. Neither is Dennis Bamford, who is well known for thinking about structures, that tree we looked at for the double beta barrels, for viruses all being related to each other, Finnish professor. This fellow, however, Antonio Alcami, is a member of the WHO Advisory Committee on Variola Research, and he's a monkeypox researcher in Spain, which is where I went to a meeting, the Spanish virology meeting, and then we got to go to this place called Atapuerca, which is just outside of Burgos in central Spain, which has a really amazing archaeological record of various different prehumans for over 100,000 years. And this is the skull of one of those really ancient virologists who was studying there at the time. But the take-home message here is that variola is kept around so that we can try and find good antivirals. And there's actually an antiviral last year that was approved by the FDA for smallpox, but they say one is probably not enough. We're going to try and get another one, and so that's why they're keeping it around. Vaccination works. This is an example from the Jenner Museum. Here's a poor boy who was not vaccinated. Here's one who was. So vaccines work. Vaccines work particularly well for smallpox, but even though it was, late 1700s, near 1800, and of course now we're not moving again, there was already an anti-vax movement. So if you look carefully here, what you will notice is that Mr. Jenner, here in the middle, who is now not working, is vaccinating this poor woman here, and has clearly vaccinated other people who now have cows. Cows growing out of their arms. And hang on, go over here. Or a cow growing out of their buttocks. So this is the pock from cow. So anti-vax movements are nothing new. Even immediately after vaccination had started, there were already anti-vaccinations, and this is the wonderful effects of the new inoculation down here. So that's the older version. Here's the more modern version, clearly photoshopped. but a very unhappy baby, or at least, you know, someone actually probably is hungry, knowing anything about my babies. I like this graphic in particular. It's a little older, but I think it shows the basic message here very well. Before vaccination, after vaccination, and this is the most recent number of cases, 21,000 cases. Actually, this is morbidity, not even cases. So times that you're actually having some kind of disease, which is getting somebody into the hospital. 21,000 gone to zero. Hepatitis, 117,000, 11,000 down here. Polio in the US had 16,000 down to zero. Smallpox, 29,000 down to zero. The unfortunate one is here. You know, measles, we had 500,000 a year. Now this particular year, we're almost actually over 800, pushing 1,000. But just, I find it hard to believe that people don't like these kinds of data. But be that as it may, there are other people who've done really much better jobs talking about vaccination. I think John Oliver's discussion on vaccination is a really nice one if you want to show somebody about that. And if anyone likes John Oliver, it's very appropriately John Oliver. Wanted to now briefly switch gears and talk about, just show you a video on the process of smallpox eradication. This is talking about that whole process with the man who is really probably most responsible for this, Robert Henderson, who unfortunately passed away just 2 years ago now. And this is from the Canadian Broadcasting Corporation, the virus hunters, and I'm not quite sure how it happened, but it actually featured me as part of this. And being in a TV documentary with Robert Henderson is really a huge honor as far as virology is concerned. So let's take a quick look at this. Hopefully this will now work in terms of you being able to hear it. Let's give it a try. We'll go here.
In the world of virus hunters, this man is a giant. Dr. Donald Henderson accomplished what no man has ever done.
Donald, not Robert, sorry.
He completely wiped out a virus. And not just any virus, but the deadliest and most feared of them all. Smallpox. The virus killed 300 million people in the 20th century alone. And it left millions more disfigured and often blind.
It's been said that smallpox has killed more people than all the wars in history combined.
Donald Henderson is still haunted by memories of visiting the victims.
Their faces were anxious, there were flies all around, some were crying, some of them you could see were dying. The odor was just overwhelming. It was like rotting flesh. And the British physician I was with put his hands on the railing and he said, I could never do that again. I could never do rounds on these patients. That is the most horrible thing I've ever witnessed in my life.
The battle against smallpox, in fact the outright war on all infectious diseases, started in earnest at the end of World War II. It was an era of science and progress, a time of confidence, some would say of hubris.
There was a tremendous optimism that science was going to protect us and it was going to solve all our problems. Penicillin had just made its debut, and this was effective against bacteria. There was a kind of sense that we would have something for viruses soon, if not immediately, and in any case, we had vaccines.
No one doubted germs would surrender to medical ingenuity, and they did.
DDT, the miracle insecticide that helped prevent epidemics in war-torn Europe and the Pacific areas, goes to work in an American community.
It seemed like a golden age where many of the diseases we used to fear in the past, like whooping cough, diphtheria, smallpox, polio, all seemed to recede into the woodwork because we could control them.
With smallpox and other infections apparently in retreat, research could focus on the remaining killers, chronic diseases like cancer and heart ailments. These were now the biggest challenge in the war on disease. In 1954, as a newly minted doctor, Donald Henderson was ready. It was a time of optimism. It was a time of delusion.
These were great triumphs of public health and great triumphs of science, having these vaccines controlling these once feared and once deadly diseases. But what really happened was sort of more out of sight is out of mind. They were gone only in our thoughts.
Bradford faces a grim situation.
In reality, outside of North America, smallpox was very much alive.
In the early 1960s, there were many importations of smallpox into Europe. Part of the reason was there were big outbreaks in India and Pakistan, and they were coming across as visitors or immigrants and starting outbreaks. And I was concerned that it was going to happen in the U.S., and we hadn't had any smallpox for so many years.
The world's wealthy nations, where smallpox was a fading memory, now had to face a stark truth. They would never be safe until the virus was destroyed everywhere. In 1966, the World Health Organization selected Donald Henderson as the man to do it. He was under 40, old enough to lead, yet young enough to believe.
We were engaged, if you will, in a crusade to do something that had never been done before, that could be done.
When the global eradication program started, there was smallpox in 43 countries, 15 million cases a year. Vaccine was scarce, and there was never enough money. All the team had was heart.
They were young, they were willing to spend incredible amounts of time in the field, they worked very hard, and let's say that they lived from the field. They were not very good at embassy cocktail parties.
The virus had one fatal flaw. It existed only in humans. Since no animals carried it, if every human at risk could be vaccinated, smallpox could be beaten. In the first year of the program, Henderson's teams vaccinated over 25 million people on 3 continents.
Smallpox is by far the easiest of all the diseases to eradicate. You need to vaccinate only once, and the individual is protected. You had a vaccine that was very stable. You knew where the smallpox was all the time because everybody who could transmit the disease had a rash, and a very typical rash. There were no silent carriers who could spread it.
But teams still had to go out and hunt it down. A photo of a baby with smallpox said everything people needed to know to help find the victims. Slowly, one needle at a time, the killer was cornered. By 1977, smallpox survived in just one country in the world, Somalia. And then in just one person. Ali Mao Mellon, a 23-year-old hospital cook in America, was the last natural case of smallpox in the world.
It would, ironically, be a case that should never have happened.
Sorry about that.
Here's a young man who had been vaccinated, but he never got himself vaccinated. And then he came down with smallpox, and by this time he had contact. I think the count was about 110 people.
Now it was a race against time. A frantic vaccination campaign hit the streets. That one last case meant almost 55,000 people had to be vaccinated, and fast, to avoid disaster. Henderson and his team beat the odds.
It was the first time in all of recorded human history that all of the species we have driven to extinction actually manage to drive an infection, an infectious disease, to extinction intentionally.
2 years later, the world was officially declared smallpox free.
The end of smallpox should not be viewed only as the closing of a magnificent chapter in international collaboration. It should also be seen as one of the launching platforms for our goal for health all by the year 2000.
The year 2000.
How did we feel? I suppose, as people said, you get to the top of the mountain, and it's wonderful. But the fun was in there. And I think we all realized that we spent a lot of time in the field. We had a lot of good friends. Lived through a lot of bad times together. And it was all over.
Donald Henderson's victory was a spectacular exception. Smallpox is still the only virus in the world that's been completely wiped out. The war on infectious diseases had really just begun, and the deadly new viruses would be much more stubborn.
So, yeah, that was a couple of years ago. I'd say, unfortunately, Donald Henderson passed away about 2 years ago. I think the parallels with polio and the parallels with measles are also really striking in terms of, fewer and fewer places where you find them, fewer and fewer people who have them, but still lots of importations that are happening. So if you really want to get rid of the disease, you have to completely eradicate it throughout the world. And unfortunately, where was the last place where there were naturally occurring smallpox? Somalia. And that was actually when Somalia was relatively stable politically. Of course, now anything but. So again, a lot of geopolitical issues above and beyond just the fact of having good vaccines. But still, that being said, really amazing was that we were actually able to, we, of course, being the royal we as well before I get involved with viruses, we're actually able to completely get rid of smallpox, although again, 2 different places where they are still working on smallpox and still working on getting smallpox antivirals. Part of the reason for that, of course, is because we now have synthetic biology and people can start to make these viruses. Turns out it's still very, very difficult to do so. There was a group that published a paper on horsepox virus, which wasn't even on that list that we had earlier. It took them about 10 years to actually make a functional, active horsepox, and a lot of that actually has to do with the fact that the virion itself, if you just put the DNA inside a cell, it can't work because it needs all of these extra viral proteins. And so we'll talk more about that a little bit later on. So if anything, polio is much more of an issue to be synthesized because it's positive strand RNA. All you need to do is get the RNA inside the cell, it will make a virus. Whereas for these pox viruses, it's a very different story. Is everybody happy with the video? Can answer a question on it? Yes? Should be a straightforward one, hopefully. So, last known person to have a case of naturally transmitted smallpox was a vaccinator, a doctor, a nurse, a butcher, or a biology professor. You haven't clicked yet? Click. 3, 2, 1, stop. Someone thought butcher. Was a cook, yes, but not a butcher. But yeah, vaccinator. That was the main problem because this person had been together with lots of people who could then potentially come down with smallpox. So that was why they had to have that massive extra vaccination campaign right at the end. So let's talk a little bit about the molecular virology now. The Vaccinia genome, first just in sequence, as I mentioned before, it's not obviously a cow virus. Cowpox has been isolated and completely sequenced, and Vaccinia doesn't really look like it. It looks as much like smallpox as it does like cowpox. So exactly where it came from is not entirely clear. 100 plus proteins encoded in the genome. Many of them are present in the virion as well. And we'll look at some of the specific proteins, particularly those involved in replication and transcription a little bit later on. But the virion is just loaded with proteins. And again, that's partly why it's not so much of a biodefense risk. That virion is really big, 300 by 200 by 150 nanometers. I haven't printed this one out yet, but even at a million-fold scale, it would be probably about the size of my fist, so definitely larger than any of the virions we've talked about before. The genome itself is also the largest one that we've talked about so far. It's about 200,000 base pairs. In this base pair, the 200,000 base pairs, we have inverted terminal repeats, just like we had with adenovirus. Not surprisingly, these inverted terminal repeats are probably important for replication, just like they were for adenovirus. And the very ends of the genome, these are also non-coding sequences. But it's a very small percentage of the whole genome. So if you think about something like the human genome, depending on how you count it, but it's either repeated sequences or introns, non-coding DNA. Protein coding DNA in our genome is only about 1.5%, at least of the unique sequences. Here it's about 98% coding. So very different from that point of view. So let's look at these genome ends in a little bit more detail. So this, again, is just the overview of the genome for Vaccinia, about 200,000 base pairs. And at the very end of the genome, there's this loop. And so I mentioned before, this is very strange kinds of structure. Instead of having, say, just an end, where you have like a protein attached to it, again, as you see in the adenoviruses, this strand is completely contiguous. So if we're going from 5' to 3', it goes C, T, 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 A, G, T, T, A. So just completely contiguous sequence all the way around the end of the genome. And that's true at both ends of the genome. So this end of the genome and that end of the genome. And these are completely identical to each other. Again, inverted terminal repeats. So this sequence here is the same as this sequence on the other strand over here. So these looped end structures. Now this is great because there's nothing for your exonuclease to chew on. 5' to 3' exonucleases, 3' to 5' exonucleases. Exonucleases is completely protected. But then the question immediately comes up: how do you start to replicate these kinds of genomes? So we'll see that in just a second here. What are the proteins that are doing this? They're all mixed up in the genome. So remember, I think David pointed this out nicely, early genes, late genes, etc. Here, they're all mixed up in between each other. So here's a classic structural protein. It's the core protein. We'll look at that a little bit later on. This would be a late protein, but it's right next to early proteins, and then you've got core proteins that are made early and late. So these guys are all mixed in with each other. So there's no obvious, one part of the genome are as early as one part of the genome is late. This is a complete hodgepodge of different things. And as we'll see, the way that works is because they're regulated at a transcriptional level by different transcriptional regulators. So they'll have early transcriptional regulators, late transcriptional regulators, etc. What is this genome packaged in? It's packaged in this, people say brick-like structure. I'm not sure if I really like the brick. This is one of the best EMs here. Has a core structure right in the middle, an envelope around the outside, but also lots of proteins that are between this envelope and core structure. So again, not just genome, but really, really packed with proteins. And one of the things that Donald Henderson mentioned in that video is that the vaccine is incredibly stable, which is one of the things that allowed us to eliminate smallpox. These are enveloped viruses, but I kind of use quotes for the enveloped because they're so packed with protein, both in the membrane and inside the membrane. They're incredibly resistant to heat, to drying, etc. That's one of the reasons that it's so infectious. And I'm not sure if this is actually true, but apparently in the Middle Ages, what people would do if you were besieging a city and someone died of smallpox, you put them in a catapult and flung them into the city. And that would be a way that you could infect people, just because all of the various virus particles were so stable, which is great for a vaccine, but really bad for transmission of disease. So that's the one of the forms. Interestingly enough, poxvirus has actually formed two forms. This intracellular mature virus that has a single membrane around the outside, but then there's some extracellular viruses which have two membranes around the outside. And this extracellular enveloped virus seems to be important for getting from cell to cell inside one organism. So if you think about it, what that external membrane is going to do, that then will be able to fuse with a new membrane. But if you want to have a stable form, you just have one membrane. So this is the form that seems to infect from organism to organism, and this goes from cell to cell in one organism. Again, in the case of smallpox, this would just be humans. So you have these virions with the genome and lots of proteins. How do we have our replication cycle? And this is I think, potentially the most interesting or weirdest part about these poxviruses. They're DNA viruses, but they're DNA viruses that replicate in the cytoplasm and not in the nucleus. And that immediately tells you that they've got practically everything they need in that virion, and they don't have to go to the nucleus. All the other DNA viruses we've talked about, and even some RNA viruses like influenza, those have to go to the nucleus. And for influenza virus, why is that? You go to my list, but why does influenza virus have to go into the nucleus? Seal caps, and that's where the caps are. So what does that tell you about the caps on Vaccinias? They're some way of the Vaccinia making their own, or they're cap independent, one of those two options. So this cytoplasmic replication process is really fascinating. So you have first the virus gets inside the cell, and it's got that single membrane on the outside, seems to fuse at the plasma membrane, release not only the core, but also all of these other proteins that are associated with it. And so all of these other proteins are involved in starting to make genome, but also starting to make the early messenger RNAs. So again, in the absence of any nuclear proteins, you have all the proteins you need to make messenger RNA to start to make these early proteins. And by the most important of these early proteins are these VITFs. What the heck is a VITF? So a viral intermediate transcription factor. So here are the stages of how, I should have mentioned this before, we've got early genes, intermediate genes, and late genes is what they call them here. So the early ones are going to be involved in making more of the genome and also making the proteins that you need to make intermediate proteins. These intermediate proteins are going to be important for making your late proteins, which are your classic structural proteins. Those then will lead to making virions, and these virions will get assembled either as a single enveloped particle or a doubly enveloped particle. And again, the singly enveloped is the one which seems to go from organism to organism, and the doubly enveloped going from cell to cell, which we'll see going on in the process here. So one of the things to mention, again, with these early proteins, Early proteins include the proteins that are important for making intermediate genes. So here, early proteins, intermediate transcription factors. Intermediate genes, late transcription factors. Late proteins, early transcription factors? Why are early transcription factors being made by late genes? Exactly. You need them back up here. So it's just one step ahead, and these early transcription factors get packaged into the virion so that when they come in, you can use them for that process. So let's look at some of those packaged enzymes. Basically, everything you need to make messenger RNA is present in the virion. So RNA polymerase, these early gene specificity factors, those are VEGFs. Capping proteins, guanylyl transferases, ribose methyltransferases, just like you would see with cellular processes, guanylyl transferases, ATPases, poly-A polymerases, RNA helicases, DNA helicases, protein kinases, basically you name it. These are the things which are packaged inside the virion. Then they will make, as some of their, proteins later on, these now are going to be your, classic early genes for making genome. They've got their own DNA polymerase. They have their own processivity factor, topoisomerases, single-stranded DNA binding proteins. Basically, you name it, they're here. But one very important, some of the sense, difference has to do with this one right here. So DUTPase, uracil DNA glycosylase, thymidine kinase. Why do you think these are so necessary? Where is this virus replicating? In the cytoplasm. Where is normally DNA synthesis taking place? In the nucleus. So DNA and DNA precursors are almost all going to be in the nucleus. Cytoplasm has what in it in terms of nucleic acids? DNA is present in the nucleus. What's out in the cytoplasm? What's getting translated? RNAs. So if you can take the RNA and turn it into DNA, and the protein that does that is a ribonucleotide reductase, because it takes that OH off of the RNAs, you can convert the RNAs in the cytoplasm to deoxyribonucleotide triphosphates and then use them. So completely independent of the nucleus because it's using the RNA present in the cytoplasm in order to make its genome. And of course, it uses the RNA precursors to make the RNA, again, through its own RNA-dependent RNA polymerase. Okay, so just reminder here, early transcription factors, as Melissa pointed out here, in the virion, the intermediate transcription factors are early genes, the late transcription factors are the intermediate, and then we have these guys being made after replication, again, getting them into the virion, and everything is happening in the cytoplasm. Which again, to me, is really amazing that you have these viruses which can then still replicate in the cytoplasm. They still need ribosomes. So that's that last thing that viruses still haven't figured out how to do themselves. So translation, they can tweak translation, but so far no one's found any ribosomes encoded in viral genomes. That's even true for the giant viruses that we'll talk about a little bit later. I need a breath. So what does that mean? We'll ask you a clicker question. VETF proteins in Vaccinia virus are expressed as which of the following genes? Let's turn this on. Immediate, early, early, intermediate, late, or very late. Start. Let's get 100% again. 96 was close for the last one. 10. For, okay, we do not have a consensus yet, so let's have a quick discussion. What do you vote for and why?
I'm assuming that these are vesicles.
We'll get to that in just a second.
Okay. But the vesicles aren't everywhere.
It's not even really vesicles, so yeah, we'll talk a little bit more about it. Just a sec. Yeah, yeah. We'll get there. Okay, let's go again. We have 24, 25 that are our max last time. Again, feel free to continue to discuss if you like. Does anyone else want to vote? Going once, going twice? We'll just stop it because we're at 100%. So, yes, D is correct. Early, intermediate, and late are the three that we have. I needed two more, so I made it intermediate, early, and very late because those don't actually really exist. Always so much fun to try and come up with different answers for these things. So, sorry, let me close this. So let's talk a little bit more about how this transcriptional regulation works. Mentioned before that the RNA polymerase, so DNA-dependent RNA polymerase itself, which is supposed to be this purple thing down here, is very similar to the eukaryotic RNA polymerase II. Multiple subunits, seems to recognize DNA, has the standard procedures. One thing that's different, however, is that the promoters that are recognized by Vaccinia virus polymerases don't have obvious Tata boxes. They just have these promoter sequences. And these promoter sequences are specific for the early, intermediate, and late genes. And those are the ones which are recognized by the early transcription factors, the intermediate transcription factors, and the late transcription factors, together with the cellular general transcription factors. But just these individual ones are good enough. And they bend the DNA just like you would expect to see with the Tata binding protein, TF2D, etc. And once these messenger RNAs are made, guanylyl transferase puts on a totally regular cap structure, and the ribose methyltransferases put on methyl groups to give you a completely normal cap structure. These cap messenger RNAs are then completely normally translated. And all of the regulation in terms of the RNA production is going to be these, promoters, early transcription factors, intermediate transcription factors, and late transcription factors. So that's actually pretty similar to what goes on in cells. The replication of the genome, on the other hand, is very different because we don't have a DNA primase. These have very strange-looking end structures, and so that's just drawn here as a loop at the end. So how do you get replication, to start here? The DNA polymerase is a viral DNA-dependent DNA polymerase, but it's just like all of the cellular DNA-dependent DNA polymerases. It needs a template, it needs a primer, etc. So how does that happen? What happens is you have a endonuclease, it's a viral endonuclease, that cuts near the end of this sequence. And remember, this is the sequence that can form a hairpin. It's got that extra piece, but it's not completely base paired. So what happens here is it cuts, and there's actually also a helicase, DNA helicase, again, encoded in the viral genome, which will separate this single strand from here. Now you've got a template, and you have a 3' OH. So DNA polymerase can extend out to the end. Now, that's just between where this nick is and the end that you would have out here. But these sequences are complementary to each other. So what they can do is they can now base pair back to themselves, and you have a 3' end here, which is next to this piece of DNA. And again, this has a DNA helicase, so this DNA polymerase will extend. It extends all the way down here, around the other end, and all the way back out to this end. We have a DNA ligase that hooks those two ends together. Then you have some kind of resolvase, actually not unlike a holiday junction resolvase, where you end up separating these two genomes. This can start again, making a nick, extend to the end, etc. So this is actually self-primed replication, which I think is really smart, again, to totally over-anthropomorphize these viruses. So they don't need a primer. They actually have their own primer in their genome. And as we'll see when George talks about the parvoviruses on Friday, these single-stranded DNA viruses, at least these particular ones, basically do a very similar thing in order to replicate their genomes. So this is the replication process. Making messenger RNAs, the early messenger RNAs, classic process where they're making their normal capping, tailing, etc. Curiously, some of these late genes actually have 5' sequences, which are poly A's and are made by stuttering. Actually, not unlike what you see in some of the RNA viruses. Very bizarre. We don't quite understand how this works and why it works, but if you look at those, you do see this. And then 3' ends, you've got a lot of termination read-through that happens as well. So the late RNAs in these poxviruses seem to be a little bit different than the early and the intermediate ones. They still got translated. They still seem to make plenty of their genomes. But then you have late messenger RNAs. Those get translated. It gets translated into viral proteins. How do those viral proteins start to get put together into virions? And so this is the question that Echo was asking during the last clicker question. So in the cartoon, we had these regions with these sort of crescent-shaped particles in them. And that's actually exactly what you see if you look at a cell which is being infected by one of these poxviruses, particularly Vaccinia. Have lots of these structures here which kind of look a little bit like membrane vesicles. This is where the virus assembly takes place. And also, it's a part of the cell which, if you just look at the cell, you don't see any of the regular organelles here. The nucleus has gotten pushed away, the endoplasmic reticulum gets pushed away, the Golgi gets pushed away, and all you see are these replication intermediates. This is also known as the viral factory. So if you look at one of these Vaccinia-infected cells, and we talk about giant viruses later, you'll see exactly the same thing, is where you have the replication is taking place and where all of the virion assembly is taking place. So it's a separate, almost like a separate organelle that forms for the production of virions. So your virus factory, also you can call it a virion factory, where these are being produced. Once you have all of these, and this is the final mature virus particles right here, these then need to get outside of the cell. They actually will leave the virus factory and then go to the Golgi. And in the Golgi, that's where they pick up their single membrane. And this would be, again, the one which, when cells die, that can be transmitted to other organisms. But then if they're going to transfer from cell to cell, they end up picking up yet an extra membrane here. This extra membrane, in fact, it's an extra double membrane, will fuse with the plasma membrane. And now you have a double membrane formed virion, which can go out and infect other cells. And this process of going out and infecting other cells is a really, again, I think, interesting one. Because what happens is some of these proteins that are present on the membrane of these Vaccinia viruses are really good at polymerizing actin. So what happens is if you have these virus particles with these surface structures, actin filaments will form behind them and literally push these virions out of the cell toward another cell. These are also called rockets. The enveloped extracellular virus, these then are attached to actin tails. And these are, in a normal cell, we have here normal actin filaments. In a Vaccinia-infected cell, you can see some of these actin filaments with the virions attached to them. If you blow up some of these, the little red spots are the virions, and the actin filaments are formed behind this. If you look at this in the electron microscope, here's your virion at the end, and here's that actin filament. And if we switch over here really briefly, we can actually see a video of some of these things happening. Let's move this over. So here, if it will actually load, you'll have now in fact it's flipped. So the green are the Vaccinia and the red are the actin filaments, which I was hoping it would actually load. It seems not to be completely loading here. These, well, you've got the link. You can take a look at it later, where you literally can see the actin filaments forming and pushing these virions. And why might this be happening? Well, if you think about it, in most multicellular organisms, you've got cells right next to each other. And so presumably it's the Vaccinia-infected cell making those particles. It's the production of these actin filaments that basically push that virion into the next cell. So it's a way of getting, cell-cell infection by polymerization of actin. Curiously enough, the Listeria bacteria do exactly the same thing. They have a protein which stimulates actin polymerization that also helps them move around inside of cells. So this is not loading, but I'll stop here. Anyway, again, on Friday we'll have a guest lecture. It'll be George Kaysen will be talking about single-stranded DNA viruses that are infecting animals.